All right, welcome to our explainer. Today, we are diving into something that on the surface sounds like it's straight out of science fiction. We're talking about a hidden and, frankly, a really deep connection between CRISPR, the revolutionary gene editor, and the AI that powers things like ChatGPT. So, it all starts with a question that, let's be honest, sounds a little crazy. How on earth is a tiny biological machine editing a strand of DNA anything like an artificial intelligence writing the next word in a sentence? Well, stick with me here, because it turns out the fundamental logic they both use is shockingly similar. And look, we're not just talking about some flimsy, surface-level metaphor here. This is a deep operational parallel. As our source material points out, the core process in both of these wildly different systems follows this exact same pattern. A guided search for something, a moment of recognition, an activation step, and then a final, precise action. This shared logic is the key we're going to unlock today. Okay, so to really get this, we've got to start with the biology. We need to ground ourselves in how this actually works inside a cell. So let's kick things off by unpacking the elegant step-by-step logic of the CRISPR-Cas9 system. So here's the first thing you absolutely have to get. Cas9 is not just some blind pair of molecular scissors snipping away at random. No way. It's an incredibly sophisticated little machine that runs a very specific, guided, and conditional program. It only, and I mean only, acts when all the right conditions have been met.
So how does it pull this off? Well, it's this really elegant 5-step dance. First up, it loads what's called a guide RNA. You can think of this as its search query, the thing it's looking for. Second, it zips along the DNA, scanning for a tiny 3-letter tag called a PAM motif. This is just a quick filter to see if it's even in the right neighborhood. Third, if it finds a PAM, it then carefully tests the DNA right next to it to see if it perfectly matches its guide. Fourth, and this is cool, if the match is good enough, the entire protein literally changes its shape to activate itself. And finally, step 5, it makes a clean, precise cut.
You see that pattern? It's a pipeline. You search for a target, you confirm a match, until you cross a certain confidence threshold, you activate the tool, and then you perform the action. Search, match, activate, act. Remember that pipeline, because we're about to see it pop up in a place you'd never ever expect. All right, let's make the jump. We're leaving the wet world of biology and heading into the dry world of silicon. We're going to see how that exact same 5-step logic shows up when a large language model is figuring out what word to say next. Step 1, the search template. For Cas9, that was the guide RNA, right? It defined the physical DNA pattern it was looking for. Well, for an LLM, your prompt, everything you've typed so far, gets turned into something called a context embedding. It's like a complex mathematical fingerprint that defines the meaning it's looking for. So both systems start with a target, one is just physical and the other is semantic. Step 2, filtering the search. Remember how Cas9 uses that little PAM motif to quickly ignore huge chunks of DNA? It's a super efficient filter. An LLM does pretty much the same thing, but it uses something called attention mechanisms. It looks back at all the previous words and filters for only the most relevant ones to pay attention to. The point? Both are brilliant ways to cut down the noise and focus only on what matters. Okay, step 3, verifying you found the right thing. And this is where it gets really fascinating. Cas9 uses actual physical chemistry, base pairing, to feel its way along and confirm a sequence match. An LLM uses pure math. It checks for vector similarity to find the closest semantic match in its vast conceptual space. You see the parallel? One is chemical, the other is mathematical, but the logic, find the best fit, is identical. Step 4, activation. Once Cas9 is confident it has a match, it physically reconfigures itself. It changes shape to get its cutting tools ready. Now, in an LLM, as it zeroes in on a likely answer, the information flows and transforms through many digital layers. With each layer, it's refining its prediction, basically building confidence until it's ready to make a choice. It's the same buildup of certainty, just in a different medium.
And that brings us to the final step, number 5, executing the action. Cas9 makes a precise, irreversible cut to the DNA. The LLM emits the next word. Both are committed actions. It's the point of no return. Once it's done, it's done. You can't uncut the DNA, and you can't unsay the word. Uncertainty is resolved, and the system moves on.
Okay, we've been deep in the weeds, comparing the mechanics. Now it's time to pull the camera back and look at the whole forest. What does this stunning parallel tell us about the nature of intelligence itself, whether it's grown or built? And here it is, the whole thing laid out side by side.
It's kind of wild when you see it like this, isn't it? The guide RNA in the cell is like the prompt for the AI. The PAM filter is like the attention mechanism. Chemical base pairing is like mathematical vector similarity. It's a direct one-to-one mapping of operational logic.
But of course, no analogy is perfect, and it's really important to see where it breaks. Cas9 is playing with real, physical stuff. Chemical bonds, energy, molecular shapes. LLMs live in a completely abstract world of statistics, probability, and high-dimensional math. So the physical how is totally different. But the what, that core logic, is the same.
And this leads us to a pretty profound idea. What we're really looking at are two different flavors of intelligence that have arrived at the same fundamental solution. On one hand, you have biological intelligence, which excels at molecular pattern recognition and physical search. On the other, statistical intelligence, which excels at, well, statistical pattern recognition and semantic search. But at their heart, both run on conditional logic.
And this is the thought that should really give you pause. This elegant architecture, this logic of guided search, of building confidence, of conditional action, it's not some brand new thing we invented. Nature figured this out through evolution billions of years ago. We've only just rediscovered its blueprint and built it out of silicon.
So, after all that, what's the one single crucial idea to take away from all of this? What's that golden thread that ties the living cell and the thinking machine together?
I think this one line from our source material just says it perfectly. Cas9 acts when its certainty about a physical pattern gets high enough, and LLM acts when its certainty about a semantic pattern gets high enough. At the end of the day, it's all about one thing: taking decisive action once you're confident enough in what you found.
And that really is the mind-bending conclusion. It's the same exact logic for turning information into action, just playing out on two completely different stages. One is the carbon-based world of biology, the other is the silicon-based world of computers. It seems that whether it's through billions of years of evolution or a few decades of engineering, really smart systems tend to find the same elegant solutions. Thanks for joining us for this explainer.